Okay, hello everyone. My name is Salma Metwally. I'm a second year medical student, and today we will talk about cholesterol and steroid metabolism. Okay, so our objectives for this lecture will be about cholesterol and cholesterol esters, cholesterol metabolism, regulation of metabolism, and some related diseases. We will talk briefly about bile acids and bile salts, and then lastly, we will end by talking about steroid hormones and again, some related diseases. Okay. So for the first objective, cholesterol and cholesterol esters. So what is cholesterol? It's basically a lipid molecule, all right? And you don't really need to know the structure, just know some basics. So we have 4 rings and we have 27 carbons. It has a side chain, and this OH is important for esterification, and we will talk about this on the coming slide. But you don't really need to bother yourself with the structure. Now, some functions of cholesterol are that it's part of the membrane, so cell membrane, for example, and it's a precursor to very important molecules. And some of those are bile. So specifically in bile, it will give you bile acids and bile salts. And it also gives you vitamin D. There's a whole process and it has to do with the kidneys and all that. And lastly, steroid hormones like androgens, estrogens, corticosteroids, and glucocorticoids. And we will talk about these at the end of this lecture. Now, what is cholesterol ester? So when we say ester, we basically mean that we're adding a fatty acid onto it, okay? So, for example, fatty acid has a carboxyl group and cholesterol has a hydroxyl group, which we said is over here, okay? Now, if we form an ester bond between the carboxyl of the fatty acid and the hydroxyl group of the cholesterol, we will form a cholesterol ester. So now we have esterified cholesterol. And we have 2 main enzymes that do these for different purposes, okay? So we have LCAT and ACAT. LCAT is for HDL formation. ACAT is for storage. And basically, when we esterify cholesterol, it makes it more hydrophobic. And we need it to be more hydrophobic for 2 main reasons. First one is to transport it. The second one is to store it. So when we esterify it in order to store it, we're using this enzyme, ACAT. But when we esterify it for transportation, we're using LCAT. And we know that, I'm not sure if you took this lecture, but generally when we transport lipid molecules in blood, we need to place them in something called lipoproteins. So for example, over here is an HDL, which is a lipoprotein used to transport lipid molecules like cholesterol in blood. Now, I want you to look at these pink balls and the orange balls. So the pink balls are cholesterol esters and the orange ones are unesterified cholesterol, so without this fatty acid attached to it. If you notice that the esters are inside the ball, but the unesterified ones are on the surface. Now, if you want this HDL to flow smoothly in blood, we need it to be compact and packed well. So we esterify the cholesterol in order for it to be pushed into the core of the HDL and make it spherical and more compact because it's more hydrophobic. Since the unesterified ones are less hydrophobic, they can go near the surface because around here you have water. Okay? And again, we store cholesterol in the form of the ester. All right. Now, where do we get cholesterol from? First source is the diet. So we know that when we eat something that has lipid in it, we absorb it. After digesting this lipid, we absorb all the lipid products as chylomicrons. We also know from the GI block that the lipase enzyme will digest triglycerides. And all you're left with in the chylomicron after the lipase does that would be cholesterol. The lipase doesn't really digest cholesterol that much, so this will remain in the chylomicron remnants. What kinds of foods give you cholesterol? Animal products, and eggs are a good source of cholesterol. Now there's this point, which is lowering cholesterol in the diet has very little effect on blood cholesterol, and it's mainly what we synthesize. What do we mean by that? So for cholesterol, we have a balance. We have cholesterol coming in and going out. For the one going out, if we assume, these numbers are all assuming. So if we assume that 1,000 milligrams is excreted in bile, which is, by the way, the main method by which we excrete or get rid of cholesterol. Let's assume that we excrete 1,000 milligram. Now, when we look at what we take in, assume that you eat 400 milligrams, which is the average that you get by the diet. And as a rule, you always absorb 50% of the cholesterol that you eat. So if you eat 400, you're absorbing 200. And let's assume that we make in our body by de novo synthesis 800 milligrams of cholesterol. So the net that's coming in would be 200 plus 800, which is 1,000, and out would be 1,000. So we have a balance. Now back to our sentence, lowering cholesterol in the diet has very little effect on blood cholesterol, and it's mainly what we make or what we synthesize. Because if you look at this, we make 800 compared to the 200 that we absorb. It's much more than what we absorb, right? So if we really want to like make a change in the cholesterol in the blood, we need to target the synthesis. Obviously, you know, the diet has an effect, but there's a larger effect by the synthesis. Okay. Another source of cholesterol other than the diet would be extrahepatic tissue. So, you know, fat is stored all around the body. And when we want to get this fat out of these tissues and back to the liver, we use HDL, which is our lipoprotein that we talked about. So HDL transports lipids, including cholesterol, from the peripheral tissues back to the liver in a process called reverse transport. And the enzyme that we use to esterify cholesterol in order to place it into the HDL is LCAT, which we talked about over here. And by the way, this is what it stands for, and this is what ACAT stands for, all right? Okay, so again, LCAT places the cholesterol in HDL to send it to the liver. There's also IDL, which is intermediate lipoprotein. And when you study the lipoprotein lecture, you will know all about this. Now, the last source of cholesterol is the de novo synthesis, which is basically the biosynthesis of cholesterol. And this mainly happens in the liver. And there are other places where it happens, and we will see those. But it's mainly liver. Okay, this is our first checkpoint. Any questions so far? Okay, great. I have a question for you, so try to solve it. You can answer in the chat. Okay, so the answer is actually A, which is converting cholesterol to cholesterol esters. Remember, LCAT is one of these two enzymes that will esterify cholesterol. So we have LCAT and ACAT. And when it does that, it places the cholesterol in HDL. It doesn't transfer it from HDL to VLDL. It puts it into HDL. It's kind of the opposite of this. This option says that it removes it from HDL, but what it actually does is that it places it into HDL. Did you get that? Okay, great. So the answer is A, converting cholesterol to cholesterol esters. All right. Okay. Next objective is going to talk about the cholesterol metabolism, regulation, and some related diseases. Okay. This part is pretty important, so I want you to focus. Okay, biosynthesis. So the definition of de novo synthesis is basically getting these small molecules and creating complex molecules from the small molecules. In our case, the complex molecule is cholesterol, and the simple molecule that we use is acetyl-CoA. So we get these acetyl-CoAs and we add them together to make cholesterol. Obviously, it's not that simple, but you know. Now, as we said, the primary synthesis site is the liver cell cytoplasm. Okay, cytoplasm, because biosynthesis generally happens in the cytoplasm. Some other sites, but you know, not as major as the liver, would be adrenal cortex, ovaries, testes, skin, and the intestine, but mainly in the liver. You don't need to bother with this equation. You just have to know the main components that go into making cholesterol, and those are acetyl-CoA, ATP, NADPH, and oxygen. And you can keep this number 18 in mind, but you know, don't bother with the rest of the equation. You don't really need to know it. So the main sources or the main components that go into making cholesterol, as we said, are these ones. So let's talk about each one. So acetyl-CoA is where we get the carbon atoms. But where do we get acetyl-CoA from? We can get it from beta oxidation of fatty acids. So when you break down the fatty acids, you release acetyl-CoA, and you can use it to make cholesterol. You can also get acetyl-CoA from dehydrogenation of pyruvate, or you can get it from the oxidation of ketogenic amino acids. So there are specific amino acids that when you break down, you can get acetyl-CoA. The next component is NADPH, and we know this from the pentose phosphate pathway. And NADPH is a reducing power. What does that mean? So NADPH is the reduced form of this. So it becomes NADP plus and vice versa, like they go back and forth. The reduced form is NADPH. What this means is that it can get oxidized by reducing something else. So when it gets oxidized, it will turn into NADP plus, which is the oxidized form. When it does that, it can reduce something else. Okay? So that's what we mean by reducing power. And other two are ATP and oxygen. And that's how we can get cholesterol. And all of these things just result from using up these ones. Okay? Now, this is very important, the rate limiting step. I cannot stress this enough. They always love to ask about it. And it's a target of many drugs, or a certain drug that we will talk about. So the rate limiting or regulated step is by this enzyme, HMG-CoA reductase. It's a very important enzyme and you have to know it. So HMG-CoA reductase, basically what it does is that, or let's talk about this step. So acetyl-CoA will be converted into HMG-CoA. Now, HMG-CoA will be converted into mevalonic acid by this enzyme, the rate-limiting enzyme, which is HMG-CoA reductase. Now, you don't have to know all the other steps. You just have to know this part in yellow. This is what you need to know. So acetyl-CoA into HMG-CoA, and then HMG-CoA into mevalonic acid by the HMG-CoA reductase, which uses NADPH. Okay? You don't have to know the steps, but you should know the metabolites or be able to identify them. So if for example, you're asked about squalene, you know that it's related to the cholesterol synthesis or isoprenoids or whatever. You know that they're related to making cholesterol, but you don't have to know the steps. You just have to know the first one, which is the rate limiting by HMG-CoA reductase. Now, degradation of cholesterol. We can't break down cholesterol in our cells generally, but we can get rid of it by just removing it. So we can release it into the feces, we can convert it into bile salts, or bacteria in our gut can reduce it to neutral sterols. Now for bile salts, this is the main method by which we get rid of cholesterol. Now, if someone has hypercholesterolemia, or in other words, a lot of cholesterol in their blood, we can use fibers. And what these fibers, like they can eat extra fibers, and what they will do is in the intestines, these fibers will bind to the bile acids and basically just carry them with them into the stool, into the feces, okay? So they will just stick to them and, take them out of the body. So now you have less bile acids being reabsorbed back to the liver. So the liver needs to make more bile acids to compensate for the bile acids secreted with the fibers. And to make the bile acids, it's going to use up the cholesterol. And you're kind of fixing this problem of too much cholesterol. Okay, now regulation. Remember, our rate-limiting enzyme is HMG-CoA reductase. It's a very important enzyme. How is it regulated? So, we have three main methods of regulating it. There's covalent modification, there is degradation of the enzyme, and there is gene expression. For covalent modification, we're mainly talking about phosphorylation and dephosphorylation, which is adding a phosphate or removing it. Generally, it's inactivated by phosphorylation, and dephosphorylation will activate it. Okay, we will talk over this again, so we'll get clear, inshallah. The second way is degradation. So basically, any enzyme or anything really has a half-life. For the case of this enzyme, it has a half-life of 3 hours. So after that, it will just, be broken down. And this half-life depends on cholesterol levels. Okay. Now, for gene expression, again, cholesterol levels control the amount of mRNA, which will get translated to make this enzyme. Let's talk more about these regulation methods. So our first one is the covalent modification, which, we're talking about the phosphate. Okay, we have two main states in our body. A feeding state, which, you just ate, and you have all these things that you ate, and you have insulin. The second state is fasting state, where you have glucagon. Now, for feeding, when you have insulin, what insulin does is it activates our enzyme here. So basically, insulin activates cholesterol synthesis, because remember, this enzyme will make cholesterol. So if you have insulin, you make more cholesterol. Because if you just ate, you have a lot of lipid stuff, like acetyl-CoA and NADPH, which is not really lipid, but you have the components for making cholesterol. And you want to put it into cholesterol. So you activate this process using insulin. How does insulin act? Generally, insulin will always induce a phosphatase, and phosphatase will dephosphorylase. So it removes the phosphate. So insulin will always activate a phosphatase. It always removes phosphate from something. In the case of this enzyme, when the phosphate is removed from it, it will be activated. Now, for the fasting, we have glucagon. Glucagon will do the opposite. It ends up inactivating this enzyme. Because if you're in a fasting state, why would you want to build cholesterol? You would want to break down what you have to get energy, right? You wouldn't want to build things. So glucagon inactivates HMG-CoA reductase in order to make less cholesterol. And it will do that by phosphorylating it. Glucagon in general, like it always, this is the pathway that happens because of glucagon stimulation. So it will activate an adenylyl cyclase, which will produce cyclic AMP, and cyclic AMP activates a kinase. Now, kinase will add a phosphate by definition. It will phosphorylate. So when it adds a phosphate onto the enzyme, it will inactivate it. Okay? So again, in general, insulin will activate the enzyme HMG-CoA reductase, and you have cholesterol synthesis. Glucagon will inactivate it, and you will have no synthesis of cholesterol, okay? It's activated when you remove the phosphate by phosphatase, and it's inactivated when you add a phosphate by the kinase, which is over here. Okay, now, we talked about the covalent modification. Let's talk about the other two methods of regulation of HMG-CoA. So, cellular cholesterol content. Remember, we have two main things that regulate by the cholesterol level. We have the half-life of the enzyme, and we have the amount of mRNA, which will give you the enzyme. Okay? Now, let's explain this image over here. So, this is a liver cell, and C over here stands for cholesterol. If you have a lot of cholesterol inside the liver cell, would you want to make more cholesterol? Obviously not, because you have too much already. So you will inhibit HMG-CoA reductase, so you inhibit the synthesis of cholesterol. Makes sense. You also will inhibit the LDL receptor, or you will downregulate the receptor. Why? Because if you have a lot of LDL receptors over here, you'll keep taking in LDL into the cell. And LDL contains cholesterol. All lipoproteins, they contain like cholesterol and lipid stuff. So if you keep taking it in, you keep taking in cholesterol, but you have too much cholesterol here. So you don't want to take in anymore. So you downregulate the receptor. And lastly, you activate something. Remember this enzyme, the ACAT, which esterifies cholesterol for the purpose of storing it, okay? So if you have a lot of cholesterol here, you might as well just store it already. So you stimulate the enzyme that stores it. So you inhibit HMG-CoA reductase, inhibit the LDL receptor or downregulate the receptor, and you activate ACAT in order to store the cholesterol. So how do you inhibit the HMG-CoA reductase? You can do that, as we said, by like downregulating the gene expression, so you make less of the enzyme, and you also shorten the half-life, so it will get degraded quickly. Okay. Isn't LDL the bad cholesterol? Why are we reducing its uptake? Okay, so it is the bad cholesterol, but in the cell, if you have a lot of the cholesterol, why would you want to take in more cholesterol? The cell itself is just like, okay, enough, don't give me more, And that's why if you have too much LDL, it will put too much cholesterol inside the cell, and you can't take it up, so it will build up in the blood. It's as a result of having too much cholesterol. And that's why having too much cholesterol is bad, because you get more of the LDL because you can't take it up. Does that make sense? Okay, great. Okay, now hypercholesterolemia. So we finished talking about the actual metabolism. Let's talk about some diseases. So mainly hypercholesterolemia is the disease that that concerns cholesterol. So let's break down this word. Hyper is high, cholesterol is cholesterol, and emia is blood. So this basically means you have a lot of cholesterol in blood. This is related to heart disease like atherosclerosis and coronary heart disease. How do you treat it? You use statins. Statins are a class of drug, and its target is HMG-CoA reductase, our rate-limiting enzyme. What it does is it decreases this enzyme. So what happens if you decrease the enzyme? You decrease the synthesis of cholesterol, okay? Which is good. You don't want to make cholesterol. You want to lower the de novo synthesis, okay? Now, how does it work? Statin itself is a structural analog of HMG-CoA. So we know that this enzyme acts on HMG-CoA. If statin is a structural analog, it will go bind to the enzyme instead of the HMG-CoA. So the enzyme is now getting statin binding to it, but it can't actually act on statin. So the enzyme won't do anything. So in this way, statins are competitive inhibitors of HMG-CoA reductase. And because they're competitive inhibitors and analogs, they bind at the active site, which HMG itself should have bound to, but it can't because statin is bound there instead. Now, when you do that, you're decreasing the cholesterol in the cell. So you're in the liver cell, and you're decreasing the cholesterol here because the cell itself is making less cholesterol. So you're decreasing all the cholesterol here. So what do you get? You get the opposite of this if you have low cholesterol in here because you're not making it. So you activate HMG-CoA reductase, which sorry, forget about that. So let's focus on the LDL receptor. This is what mainly happens. So when you're making less cholesterol, you activate the LDL receptor, you upregulate it, you place it on the cell. So now you will take up the LDL. So you're removing it from blood, which is good because the main problem, the disease, is that you have too much cholesterol. If you give this drug and you reduce the synthesis of cholesterol over here, you're gonna upregulate the receptor, the LDL receptor, the opposite of what happened at first. So when you upregulate the receptor, you're taking up the LDL from blood and you're having less LDL and less cholesterol in blood. So you're fixing the high cholesterol in the blood problem. Now, some side effects of the statins are myopathy, increased serum hepatic enzymes. I don't know how important side effects are, but keep them in mind. Okay? So again, statins inhibit the HMG-CoA reductase. As a result, you have increased LDL receptor, and you have increased uptake of LDL from blood and less cholesterol in blood. Now, this is generally. So this could happen in someone old who has a lot of cholesterol in blood, but if it happens in someone young, we would call it familial hypercholesterolemia, or FH. FH is basically due to a gene mutation, and it targets the LDL receptor. So they can't make LDL receptor really well, and this mutation is on chromosome 19. Now, if you can't make LDL receptor, you can't take up the LDL from blood. So you have a lot of it in blood, and you have a lot of cholesterol in blood, and that's why it's hypercholesterolemia. You have two forms of this mutation. There's a heterozygous and there's a homozygous. So either you get both alleles carrying the mutation, which is homozygous, or only one allele carrying the mutation, which would be heterozygous. Now, it would make more sense that the homozygous is more rare and severe, but they both cause premature coronary artery disease. So they get coronary artery disease and hypercholesterolemia at a young age. Okay, this is compared to the older age, which you would see someone old having a lot of cholesterol. This is very pretty common. But if you see someone young having a lot of cholesterol and coronary artery disease, then you might have to think about this. Okay, but homozygous, because it's more severe, there's extremely high LDL and very early symptomatic coronary artery disease, even earlier than the heterozygous. But both of them cause early, coronary artery disease. How do you treat FH? Again, dietary interventions, weight loss, exercise, but mainly you rely on cholesterol-lowering drugs, which are statins, the ones we talked about, okay? So statins in the diet will cause upregulation of LDL receptor. So remember, they have a deficiency of LDL receptor. They're making very little of it. So you need to kind of help the cell to make more LDL receptor, And you can do that using statins because statins upregulate LDL receptor. Okay. Do you have any questions? Okay, perfect. So we have some questions here. Go through them. So should I go over it? It's kind of a tricky one. Okay. B. Okay, so the answer is actually A. Now, this is referring to this part. So what does cholesterol, which of these inhibits the expression of HMG-CoA reductase gene? And we know that when we have a lot of cholesterol in the cell, it inhibits the HMG-CoA reductase. And remember this part, that the gene expression of the mRNA that will give you HMG-CoA reductase is controlled by cholesterol levels. Okay, great. Okay, next question. Yes, perfect. The answer is D, inhibit the enzyme HMG-CoA reductase, because they're asking you about statins, and that's exactly what they do. Okay, so D. All right, last one. Are you sure? The answer for this question is yes. Okay, perfect. So it is A, abnormal LDL receptor. They're asking about familial hypercholesterolemia, which is a gene mutation that causes LDL receptor deficiency. So the answer for this would be A, abnormal LDL receptor. Okay. Next objective is about bile, and it's very brief. So we have bile acids and bile salts. You may already know about bile. So it's basically this watery mixture of phospholipids and bile salts, conjugated bile acids and all that. And it's made in the liver, stored in the gallbladder, and then secreted in the intestine in order to emulsify lipids, to digest them and absorb them. So, and as we said, it's the major way by which our body excretes cholesterol. Okay. Now, when we have cholesterol and we're making bile acids and bile salts, we have three main things that it makes. In order, it would be primary bile acids, conjugated bile acids, secondary bile acids. What are the difference between all of these? So primary bile acids is what you get first when you have cholesterol in the liver and you want to make bile acids to put into the bile. You use this cholesterol and you put it into hydroxylation by hydroxylase enzyme, side chain cleavage, to remove some carbon atoms, because you know that cholesterol has 27 carbons and bile acids has 24 carbon atoms. So you want to remove the side chain. And also it goes through the cytochrome P450 oxidase system. So cholesterol undergoes hydroxylation, side chain cleavage, and the cytochrome P450 oxidase system in order to become a primary bile acid. All right, now, primary bile acids, when they get conjugated, they become bile salts. So bile salts are basically conjugated primary bile acids. What do we mean by conjugation? It's forming an amide bond with glycine or taurine. So let's look at this over here. So this is a bile acid. This is a primary bile acid. And over here are bile salts. We see that here that we just got this bile acid and we added these to it. This is a glycine and this is a taurine. And when we added the glycine or the taurine to this primary bile acid, we formed a bile salt. Why are we making bile salts? Because bile salts are better at emulsification than the primary bile acids. That's basically, So again, conjugation by glycine or taurine. Now, what are secondary bile acids? Basically, after you've used these ones in the intestine to emulsify and digest the fat, you now want to recycle these bile acids. You want to take them back up to use them again. So in the intestine, we have some bacteria that modify all of these bile acids and bile salts to turn them into secondary bile acids that we can take back in. Some of these secondary bile acids are the oxycholic acid and lithocholic acid. How does this process happen? Like to form secondary bile acids, we have two main things, dehydroxylation and deconjugation. Now let's look at the difference between primary bile acid and secondary bile acid to know why we need to do these. So this is a primary bile acid. The cholic acid is a primary one. We have 3 hydroxyl groups. Chenodeoxycholic acid is a secondary bile acid. It only has 2 hydroxyl groups. So we need to remove one of these hydroxyl groups to turn it into a secondary bile acid. And that's why we do the dehydroxylation process. And then we also have deconjugation in order to, turn the bile salts into secondary bile acids. So first, if we start with this one, for example, which is a bile salt, look at it, it has 3 hydroxyl groups because it was formed from this. And then it has the taurine, which we use to conjugate it. First, it undergoes deconjugation to remove the taurine. And then it undergoes dehydroxylation to remove one of these hydroxyl groups. And now we end up with this secondary bile acid, which is chenodeoxycholic acid. Deoxy because we removed a hydroxyl group. And again, formation of secondary happen in intestine. Now, cholelithiasis is basically stones formed by bile, basically. So we have bile salts, which we talked about. They solubilize cholesterol in bile. In the bile, the mixture of bile itself, you do have some cholesterol there. So the bile salts that you put there would solubilize the cholesterol in the bile. Now, how do you get the stones? Stones happen if you have either too much cholesterol or too little bile salts. Because if you don't have enough bile salts to solubilize the cholesterol gets too much and this excess will form stones. And again, if you have too much cholesterol that the bile salts can't keep up, the cholesterol will form stones. There's this drug called gemfibrozil, and basically what it does is it inhibits cholesterol conversion to bile acids. So you're not making bile acids. So what happens when you don't make enough bile acids? You don't have enough of it to solubilize the cholesterol in bile, and so you have formation of stones, which are cholelithiasis. This could happen like without this drug, but this is a drug that causes this as well. This could still happen if someone just has too much cholesterol. Now these stones, especially if they're small, can cause obstruction of the bile duct. And if they're large, mostly they would cause cholecystitis, which is inflammation of the gallbladder. How do you treat it? Usually they just do a cholecystectomy, which is removal of the gallbladder. All right. Another checkpoint. Is this part okay? Okay. I have a question here. Yep, perfect. The answer is E, glycine. So to conjugate bile acids and turn them into bile salts, we conjugate using glycine or taurine. Okay. Okay. This is our last part, steroid hormones and some diseases related to them. So why are we talking about steroid hormones? Because they're made from cholesterol. We have different types of steroid hormones. Those would be glucocorticoids, mineralocorticoids, and then gonadal or sex steroids. Glucocorticoids, like for example cortisol, are important for carbohydrate regulation. Mineralocorticoids like aldosterone are important for mineral balance. Gonadal sex steroids are important for reproductive functions. There are different gonadal sex steroids. Those are androgens like testosterone, estrogens like estradiol and estrone, progestogens, which are also called progestins, like progesterones. And then other than these functions, the carbohydrate regulation, the mineral balance, and the reproductive functions, we also have other functions of steroids. Those would be inflammatory responses, stress responses, bone metabolism, cardiovascular fitness, behavior, cognition, and mood. Okay. So as we said, steroid hormones are made from cholesterol through a series of enzymatic steps. And where does this process of making steroid hormones happen? It happens in mitochondria and smooth endoplasmic reticulum of steroidogenic tissues. Now, steroid hormones, because they're from cholesterol, which is a lipid, are lipid soluble and not water soluble. And this means that they can just flow through the membrane of a cell, like they can just diffuse, they're permeable to the membrane. And so we don't just make it all the time, we only make it when we need it. Because if you synthesize it, it will be immediately released. So you only make it when you need it, and it's directly released and not stored in the cell. Okay? You can't really store it because it will just escape. Now, another thing about it being lipid soluble is it can't just flow in blood because blood is water, basically. So we need a binding globulin to bind to it. And we have specific binding globulins for each steroid hormone or each group of steroid hormones. So we have corticosteroid binding globulin for cortisol, which is a corticosteroid. And we have sex steroid binding globulin for testosterone and estradiol, which are sex steroids. This last point, progestins are the biosynthetic precursor of all steroid hormones, and we will see this point again in the coming slides. We'll come back to it. Okay, steroidogenesis. As we said, it happens inside the mitochondria and smooth endoplasmic reticulum. So for the mitochondria, we need something to put it into the mitochondria. And this protein that helps us is called STAR. STAR is also known as steroidogenic acute regulatory protein. This is what transports the free cholesterol into the mitochondria. And this is our rate limiting step. Okay, very important. Rate limiting step is STAR to put cholesterol into the mitochondria in order to use this cholesterol to make steroid hormones. Now, the precursor for the cholesterol that we're using to make the steroid hormones could come from many sources. Some of these, we already know these, so we can synthesize it in the cell. We can actually make it there. We can take it from the stores that we have in lipid droplets in these cells. And we can use it, we can take it from LDL that we take up. This last point, LDL uptake, to get cholesterol for steroid hormone synthesis, is very important in steroidogenic cells, which are chronically stimulated. So if you have a steroidogenic cell that's always stimulated to make steroid hormones, it will mainly rely on the LDL uptake to get the cholesterol. Okay, actual process of making steroid hormones has a lot of enzymes, but you don't need to know all the enzymes and all this. You need to know some steps, but you don't need to know all the enzymes. There are two main enzymes that you have to know because there are clinical correlations with them. 21-hydroxylase, also known as CYP21A2, 21 because 21, and A because this is also called 21-alpha hydroxylase. So CYP21A2. And the other one is 11-beta hydroxylase, also known as CYP11B1, 11 because 11, and B for beta. And this is a general diagram of how the process goes. obviously it's more detailed than this, but this is generally you get cholesterol, you make progestins, and then from these progestins, you can get any of the other ones. You can make glucocorticoids, you can make mineralocorticoids, and you can make androgens, which you can use to make estrogens. So this is what we meant by this sentence. Progestins are the biosynthetic precursors of all steroid hormones, because that's the first thing you make from cholesterol, and then you can use it to make the other ones. Okay. This is not the same picture that you have in your slides, but I find it really nice, so I'll use it to explain. Now, we know that we start with cholesterol, and again, we have star, which is our rate-limiting step in order to put the cholesterol into the mitochondria. From cholesterol, we make pregnenolone and then progesterone. We use this enzyme 3-beta-hydroxysteroid dehydrogenase to make progesterone, which is a progestin. So now we got our precursor from which we can make the other steroid hormones. Okay, so we're at this step, progesterone. Now, there are three main pathways for this progesterone to go. First one is it can be acted on by 17-alpha hydroxylase, which is another enzyme. This will convert it to 17-hydroxyprogesterone. From the name, 17 will add a 17-alpha will add a 17. Hydroxylase will add a hydroxy group. So 17-hydroxyprogesterone. This 17-hydroxyprogesterone can then be used to make androgens. Androstenedione is an androgen. So when you make these androgens, like testosterone, you can use these androgens to make estrogens, like estradiol and estrone. So again, you get progesterone. You can convert it to 17-hydroxyprogesterone, which you can then convert into androgens that you can use to make estrogens. Okay, this is the first pathway. Second pathway is making aldosterone. How do you make aldosterone? Progesterone over here can be acted on by this other enzyme, 21-hydroxylase, which we talked about. 21-hydroxylase converts progesterone into 11-deoxycorticosterone, also called DOC. Okay? Now, DOC can be converted into corticosterone by 11-beta-hydroxylase. So 11-beta removes this 11, and then hydroxylase would remove the deoxy, because deoxy means there's no hydroxyl. So hydroxylation adds a hydroxyl and you cancel out the deoxy. Okay, if that helps you remember the name. So 11-beta removes 11-hydroxylase, removes the deoxy. So now you have corticosterone. This is the precursor of aldosterone. So again, progesterone acted on by 21-hydroxylase to make 11-deoxycorticosterone. 11-deoxycorticosterone will be turned into corticosterone by 11-beta-hydroxylase. And then corticosterone can be converted to aldosterone. Okay, this is the second pathway. Now, the last one is making cortisol. So progesterone, remember how it was acted on by 11-alpha hydroxylase, and then at the end, you ended up with androgens? Well, it can also be acted by 11-alpha hydroxylase to get the 11-hydroxyprogesterone, which we saw earlier, but then it can go somewhere else. The 17-hydroxyprogesterone can go into making cortisol if it's acted on by 21-hydroxylase. So 17-hydroxyprogesterone. If it's acted on by this other enzyme, you get androgens. If it's acted on by 21-hydroxylase, you get 11-deoxycortisol. 11-deoxycortisol will then be acted on by 11-beta-hydroxylase. And what it does, you have 11-beta will remove the 11, hydroxylase will remove the deoxy. So you end up with cortisol. Okay? So I know this is pretty confusing, but you mainly need to know this 21-hydroxylase part and the 11-beta-hydroxylase because these are related to some deficiencies. Can you repeat? Okay, I'll repeat the part about cortisol. Okay. So remember how we had progesterone was acted on by 17-alpha hydroxylase, and then you get the 17-hydroxyprogesterone. This 17-hydroxyprogesterone can be used either to make androgens or to make cortisol, depending on what the enzyme that acts on it is. If it's this other enzyme, you get androgens, and then you can make estrogens. But if 17-hydroxyprogesterone is acted on by 21-hydroxylase, you get 11-deoxycortisol, which can then be converted to cortisol by 11-beta-hydroxylase. Okay? So 17-hydroxyprogesterone into 11-deoxycortisol by 21-hydroxylase, and then 11-deoxycortisol into cortisol by 11-beta-hydroxylase. Okay, great. So now we'll talk about some deficiencies. This is the last part, I think. So we have these two main enzymes that we talked about, 21-hydroxylase and 11-beta-hydroxylase. Before we go into what they look like, let's theoretically look at what would happen if one of these enzymes is deficient. Let's start with this one, 21-hydroxylase. So 21-hydroxylase. If you don't have this enzyme, you can't get anything that's under here. So you can't make aldosterone, you can't make cortisol. You end up with these, and then they all go into this pathway, which doesn't need this enzyme. So if you have this enzyme deficient, you don't make cortisol, you don't make aldosterone, you have buildup of 17-hydroxyprogesterone, and all these will go into making androgen. So you have androgen excess. Okay, so this deficiency is autosomal recessive. It impairs the synthesis of cortisol and aldosterone, as we saw here, because you can't get down here, you can't make cortisol and aldosterone. And because you don't have enough cortisol, you get something called congenital adrenal hyperplasia. Now, I'm not sure how much you know about this, but congenital adrenal hyperplasia is basically the adrenal gland gets big. Why is that? Because normally you have this hormone called ACTH, which will stimulate the production of cortisol by the adrenal gland. And it gets secreted when you don't have enough cortisol. So if someone cannot make cortisol, they keep secreting this ACTH because the body thinks they don't, the body sees that they don't have cortisol, so they want to make cortisol. So keep secreting ACTH, and ACTH keeps on stimulating the adrenal gland. Normally, when you have ACTH secreted, it will stimulate the adrenal gland to make cortisol, and then the rising cortisol level will stop the secretion of ACTH and everything goes back to normal. But if this person's adrenal gland cannot make cortisol, the ACTH keeps on stimulating the gland, but nothing's happening. So it just keeps on stimulating, and the adrenal gland does not make cortisol. It only gets big because this stimulation makes it big. Okay? So the adrenal hyperplasia. Congenital because, it's a deficiency, so it would usually appear in, this person when they're a baby. And in severe cases, you have something called neonatal adrenal crisis. So now we know we can't make aldosterone. What happens if you can't make aldosterone? You can't retain salt and water because the function of aldosterone is to retain salt and water. So if you don't make this hormone, you can't retain them. So you have salt and water loss. And this leads to hypovolemia and shock, which causes neonatal adrenal crisis. Okay, so low cortisol, congenital adrenal hyperplasia, and aldosterone cause neonatal adrenal crisis. Now, we talked about what happens when you don't make these, but we still have all of these going into making androgens because that's the only way they can go. So you have androgen excess because all the metabolites go into making sex hormones like androgens. So what happens when someone has androgen excess? If it's a girl, there will be virilization, which is masculinization. So they will start to get masculine features. And in boys, there would be precocious puberty. So it would be early on. This is usually they're like 8-year-olds with puberty, and this is a very early age. So this is androgen excess. How do they diagnose it? There's increased plasma 17-hydroxyprogesterone. Which one was this? It was this one over here, 17-hydroxyprogesterone, because it can't go down to make cortisol. All it can do is either be made into androgens or just build up. Okay, so this can build up. And this severe case is early onset if there is neonatal adrenal crisis, but it can be late onset if it's not as severe as the ones we talked about. And this would appear as menstrual irregularity and hirsutism in young females. Hirsutism is these females, they start to get facial hair, for example, and too much hair, which are usually a male feature. So yeah, this is adult onset if it's less severe. And lastly, we have 11 beta hydroxylase deficiency. Let's go back to this. So we talked about what would happen if this green one was deficient. Now, what would happen if the orange one was deficient, which is 11 beta hydroxylase? You can't get anything that's below this, right? So you still can't make cortisol. You still can't make aldosterone, but you can make these two new things, 11-deoxycorticosterone and 11-deoxycortisol. So you still have the problem of little cortisol, and what that causes is adrenal hyperplasia, congenital adrenal hyperplasia, because again, you have little cortisol, you have too much ACTH, and you keep stimulating the adrenal gland that it gets too big, and it's called congenital adrenal hyperplasia. And because you have building up of this one, 11-deoxycorticosterone, there's something that wasn't in the first deficiency. So we know that we can't make aldosterone, but guess what? This 11-deoxycorticosterone can act like aldosterone. So in 21-hydroxylase, we talked about how there is hypovolemia, right? Because you don't have aldosterone in the first one. In this one, 11-beta hydroxylase deficiency, because you can make the oxycorticosterone, you don't have hypovolemia. In fact, you have hypertension because we said that this acts like aldosterone and it will build up too much because of this deficiency. Okay? Normally, in us, we don't get hypertension from this one, like because it gets converted into aldosterone and it's a weak mineralocorticoid. So normally it doesn't really have an effect. It only has this effect of hypertension when this enzyme is deficient and it builds up too much. And it causes hypertension because you're retaining too much water and you have a lot of volume. So there's hypertension. Okay, so high concentrations of 11-deoxycortisol, which is this one over here, that doesn't really have an effect. But you do have overproduction of 11-deoxycorticosterone, which is this one over here, and it has a mineralocorticoid effect or an effect like aldosterone, and this would cause hypertension. Even though you're not making aldosterone, you have hypertension because of 11-deoxycorticosterone. And lastly, because, you still have some of these going into making androgens like we did in the first one, because you can't really go down here. So some of them go into making androgens. You do have virilization in females and muscularization or, big muscles in males. But it's not really as big of an effect as the androgen excess that you see in this one. Okay. That's it for the lecture. Do you have any questions about the last part? Okay, great. Thank you so much for attending. I hope everything was clear. If you can scan the code and give some feedback, that would be great. And if you have any questions later on, you can contact me. I left my phone number and my email over here. And yeah, thank you.